I'm Marco again. Second slide, please. Next slide. And yeah, just to, most of you, I know the faces you are using the facility, but just in case you don't know, Tommy here is the clinic lead, I'm the city manager, and I have a fantastic team with Tom, Lucy, Gregory, and Marie. Marie, Tommy are here, also Lucy and Greg in the back.
Quick show of hands.
Sure.
You too, Tom.
And slide, please. And so I just wanted to, again, most of you might know it, but we are part of a bigger family here in Liverpool, which is called SRF, Shared Research Facilities. And in total, we are 17 facilities ranging from omics to bioimaging, NMR, you name it, that's part actually of SRF. In total, 90 staff and over 40 million of equipment. And yeah, as I say, we cover many different areas from bioimaging to multi-omics. And this was actually part of our bid in order to join a big microscopy consortium called Eurobioimaging. We can work across scales. Actually, that's one of our strengths. We can go from single molecule up to a whole animal, our mouse, actually. And the next slides want to express the same concept. So basically, we can go from the nanoscale, and that's what is actually done in the biomedical electron microscopy unit, which is led by in Prior. And here in the movie, you can see some, I think they are mitochondria, right? Yeah, they are mitochondria, I'm pretty sure, in the essential movie. And then there are many other intracellular with nanoscopy, basically resolution samples coming from EM. And this is the list of the instruments. And then we go up a scale and we go actually to microscale. What you see revolving there is a Daphnia magna for a project about plastic pollution. In the right hand of the panel, actually, I have a pointer. So here you can see a project where we are looking at the movement of colloidal particles with is a large project. That's the brain of a Drosophila. And basically, what we can do is not only image acquisition, but we have a wonderful image analyst in-house. She's Marie, and we are really helpful for anything that has to do with extracting the data from your images and try to quantify, which is actually also a big point of the Zeiss Cell. And here's some of the available system we have. We have a lot of cutting edge instruments from the light sheet. The BioFM, we don't have many of them in the Northwest. So an instrument that can interrogate your sample with atomic force microscopy, and I see some of the users here, and confocal at the same time. And we can do flame, FCS, super solution with latticine, Melara, storm, which is under super solution technique, and multi-photon. So I can tell you at national level, we are very well equipped. Next slide, please. Every step.
Sorry.
No worries. Yeah, so that's what we offer. What I say, so in addition to image, we can also do training, consultation for project, and image analysis. And going up a scale now, we are, if you want, the millimeter range, which is actually preclinical imaging, is the facility led by Harish Pokhari. And in this facility, we can interrogate the sample with a bigger scale. We can see a little mouse. We can see distribution of drugs into the mouse, presence of tumor in the mouse, live MRI. And so really, that's a strength of Liverpool that not many facilities, not many consortia of imaging have in UK. Next slide, please. And again, most of you are coming from Liverpool, but another beauty of Liverpool compared to other similar centers in UK is that everything is very cozy at a walking distance. But, I don't have to convince you about that. Please. Yeah. And yeah, probably the last thing to say, because it's pretty recent, we were actually accepted together with other facilities like in Craig and Otto Brooks and Octopus and Harwell and King's College as part of Eurobioimaging. So it's very important for Liverpool because it puts us in a consortium where we can share protocols, and in the future also we can also get funding from AU for projects, because the big point of imaging now in the microscopy community is having all the cutting edge instruments available for usage to anybody, because we have really, as I said before, like 40 million pounds of instruments, and we want them to be accessible for everybody. That's the concept of urban imaging. So the next speaker is Tom, and now we go to the serious science.
Wow. Yes. Okay.
If you need a point. Okay.
All right. So let me just. It's been doing this for a bit. Don't know what. Anyway.
All right. So, hello everyone. Those who haven't seen me around the facility, I'm Tom, the imaging specialist and one of the imaging technicians for the Centre for Cell Imaging. And I have the honour today of introducing you to our newest shiny toy in the arsenal, which is the Cell Discoverer 7. So, here it is, in residence in the facility. But what is it? So, C7 is a high throughput imaging system. So the system is designed to do imaging experiments at scale. We've got 3 imaging modes within the CD7. We've got wide field, confocal, and area scan imaging for imaging across different scales. We have inbuilt temperature and CO2 control for doing longitudinal live cell experiments under incubator conditions or other temperatures. And the system also comes with various automation options that allow us to scale up experiments in a more convenient manner. Next slide. So, when we're talking about what we can image in terms of samples on the CD7, well, with the 3 different imaging modes, we can really image across a wide range of scales. So, at the lower end of the scale, next, we have the area scan 2 imaging. So these are some images I shamelessly pulled from the Zeiss promotional material. And here we have cells infected with a bacterium within single cells, imaging at higher ends of resolution, so we can get down to subcellular resolutions of 120 nanometers with the area scan imaging. We next have more multicellular scale. Here we have kidney tubule organoids, but with confocal imaging, we can do similar scale multicellular models, which can also be combined with area scan imaging in order to enhance resolution even at this scale. And then at the upper end of the scale, we have small model organisms, be that zebrafish, Drosophila embryos, or C. elegans worms. And together, across these 3 imaging modes, we can image a wide variety of different samples. But it's not just the imaging modes that contribute to this flexibility. So the system also comes with a range of objectives from 5 times magnification all the way through to 50 times. But these can also be combined with different tube lenses. So these are optics within the system that give true magnification of the image, which come in 0.5, 1, and 2 times. So between the objectives and these tube lenses, we can really get a wide variety of magnifications from 2.5 wide field of view imaging all the way down to 100 times high magnification imaging. Now, as you'll notice, this objective is a water immersion objective, but classically with longitudinal experiments, you have issues where water will evaporate over time, especially if you're imaging at imaging temperatures. But the CD7 comes with a very clever auto immersion system. So essentially, you have a small sort of silicon membrane that sits over the top of the objective. And in there, there'll be 2 little capillaries, one that will put water on the objective and one that will draw water off the objective. So whenever you're imaging, you can switch from the water objective to the air objective and vice versa. And the system will also be able to top up your immersion medium as you image. So even though you're imaging with a water objective, doing longitudinal 24, 48, 72 experiments are possible thanks to this system. But this isn't the only thing the objectives do. So the CD7 has methods of detecting both material and thickness of the sample bottom that you're imaging through. So if you're imaging a 6-volt plate or optically thick cover slips, the system will be able to calculate that and apply corrections, just like a correction collar on an objective you might do manually. The system can actually do that for you automatically based on this detection. So enhancing the quality of these images. So that gives flexibility both on the imaging scale, but also on the mounting that you can present the sample. Now, of course, if you're imaging 50 times objectives, you still got to work with the working distance of a high NA magnification lens. But if you're imaging through plastic, it's not as great a hindrance thanks to this ability to autocorrect for sort of spherical aberrations. Now, when we talk about the next section, which is more imaging at scale, classically, when you're trying to scale up an experiment, there are three main challenges to address. The first one is how do you find and focus on the sample? Next is how you set up your imaging positions within the experiment. And the third is swapping between the samples. So each of these classically is always done manually, but if you want to upscale an experiment, these are manual steps that will add time to your experiment. So the CD7, if you go next, comes with a variety of strategies to match each of these points. So the first is the auto-focusing strategies. So these come in both intensity and reflection-based techniques. So if we go next, in an intensity-based technique, the way it works is that the system will range through the sample using a fluorescence channel, find the peak of the fluorescence in the Z, and then if you go next, it will then center your focus around that position, and then you can either do two-dimensional imaging or you can do three-dimensional Z-stack imaging with that being your reference. Very useful as a methodology if your sample is not expected to remain stationary. So if you're imaging, say, B cells, which are floating slightly above the surface, this can hopefully correct for any slight change in height of the sample. Or you have reflection-based techniques. So in this technique, next, you reflect an infrared laser across the bottom, and then the system will be able to calculate where the glass surface is and move a defined offset up. So if your sample remains in a fixed position relative to your surface, and you're looking to correct for thermal drift over time, this system will be able to correct for you. And these can be used either separately or in combination together to give you the most optimal auto-focusing strategy for finding a sample. Next, how do we sign up positions? So classically, you have the sort of sample navigator view, which this system also has. So you can either define positions within each well randomly or in a grid, or you can use tile scans to do a certain fill factor of the well. So if you want to image 10%, 20% of your well surface, you can do that. Or if you want to do something a bit more fancy, you can use the CD7's guided acquisition. So I'm going to show you a proof of principle experiment for how this works. So the first step is to acquire an overview scan of your sample. So if you're next, see here, we've got an overview scan of some PC7 cells expressing a microtubule marker. And this essentially is just to get a view of the area. Because what we're next going to do is we're going to try and identify points within the sample that are of interest. So in this case, go next again, you're trying to identify cells that had freshly divided. So we wanted to find objects that look like this. So two bright dots close together. So by using a variety of shape factors, signal, intensity, and area, we could analyze the image, identify these structures, and then the system can find objects that match that description and take higher magnification images. And what you'll end up with is something like this. So we see we've got our overview image, and then we've got our higher resolution images of the objects that have been identified. So this system can be used if you're looking for rare events. So you're looking for cells that have divided, or you're looking for cells that express a certain marker that are rare within the population. You can use this methodology so the system can find those objects for you. Next. Now, if the basic analyzer isn't enough, you want to do something a bit more complex, the system also comes with inbuilt macro scripting. So Marie, just here, has written this nice macro code for one of our users, and this can work through more complex pipelines in an automated fashion, allowing you to do large-scale analysis of all the data that comes out of these automated experiments. Now, the last thing I'm going to show you is how do you swap between samples? Well, video here. So this is a robotic arm, and what we'll see is the robot arm we'll pick up a plate that comes out the back of this incubator. It'll here. The system will identify the plate by doing a little scan barcode. So this plate is barcode, but if it had one, it'll be right there. It'll scan it to identify the plate. It'll then pick up the plate. It will now move the plate over into position. So we have the CD7 just here. It'll just nicely place that down into our loading bay. And then we'll see this disappear. And so this robot arm can both load and unload your samples. So if you're doing experiments across multiple well plates, you don't have to manually come back every time you finish one plate to then image the next one. The system will be able to automatically image your plates in a sequential manner. So this can then combine well with the other automation strategies in order to allow you to do large-scale experiments with greater ease. Now, this does all sound rather wonderful. You just stroll up to your instrument, you press a few buttons, and it basically does your entire experiment for you. And in a way, this is true. Just because it is easier to set up a scale experiment does not necessarily mean it is easy to do so. Everything still needs a level of optimization and just choosing the strategies. So you need to be able to choose the right strategies. So which auto-focusing strategy find your sample best? If you're using guided acquisition, what parameters do you need to use to find parts of the sample that are of interest to you? And these steps each need to be optimized in order to ensure that you are both accurately finding your sample and reliably finding your sample. But you are not alone. So the CCI staff are here to provide you with help and assistance, and we can provide our tools and our knowledge to help make this process a lot easier than you just budding around on your own. So with that, I'll draw an end to my talk and pass on to the next speaker.